So now I'm going for the basics of reference-based and de novo genome assembly. So reference-based and de novo genome assembly tutorials here. It involves two distinct methods, but that is de novo reconstructs a genome from scratch, while reference-based uses an existing genome as a guide. Then the tutorials for both typically begin with quality control of the sequencing reads, followed by the specific assembly method and post-assembly quality checks. So whether you are performing genome, de novo genome assembly or reference-based, your first task is to go for quality check. So all the FASTQ files, that is raw sequencing files, you need to first check the quality, and then it will cover the algorithms which are mostly used, for example, de-Bruijn graphs or overlap layout consensus, often using tools like fly, while reference-based tutorials will demonstrate mapping the reads to a known reference genome using tools like Botai2 or EWA, and then either filling in gaps or ordering contigs. So here you can clearly see the easiest difference between the de novo and reference-based sequencing are, in case of de novo, you don't know the exact whole genome of the organism, but in case of reference-based assembly, the reference genomes are easily available. For example, the ensemble Biomart database, you can check there also, but you can also check on NCBI database. So in one case, the genome reference genome is not available, that is de novo, and where you have reference genome, that is reference-based genome assembly. Now, in de novo genome assembly, you generally go for quality control, then assembly, and then post-assembly. So first you need to understand what is de novo genome assembly. It is a process that reconstructs a genome from DNA fragments without using a pre-existing reference sequence. It involves sequencing random DNA fragments, then computationally assembling overlapping reads into longer contigs and scaffolds to reconstruct the complete genome. This method is crucial for the organisms which lack the reference genome, such as novel or non-model species, and is more computationally demanding than reference-based methods. So this is very clear about the basic definition and understanding of de novo genome assembly. But the steps you need to understand are mainly three. First is quality check, then assembly, then post-assembly. So in quality check, you assess the and filter the raw sequencing reads for adapters, low quality bases, and other artifacts. In assembly, you use the algorithms to reconstruct the genome from the scratch. So for that, you have some further sub-steps, that is overlap, layout, consensus, de-Bruijn graphs, and then the tools. There are many tools, but here I have mentioned this one. But of course, I will give the details because this is just the tutorial for understanding the difference between de novo and reference-based genome assembly. Then in post-assembly, you go for polishing and annotation. That means in polishing, you correct the errors and fill in gaps, that is represented by M's in the assembled context. And then finally, annotation, where you identify the genes and other features on the assembled genome. Now, in reference-based genome assembly, the first step remains the same, but here you immediately jump for mapping, assembly, then post-assembly. So here you don't need to understand that much because it is very clear that this reference-based genome assembly involves aligning the sequenced DNA reads to an existing reference genome to identify the variations and reconstruct a new similar genome. The key steps are read alignment, which maps the read to the reference genome, followed by variant calling to find differences such as SNPs and variant filtering to remove false positives, and finally variant editing to generate the new genome. But these things depends on the goal of your project. For example, if you are working on RNA-seq, then your goal will not go for variant calling or to find the difference or to predict the single nucleotide polymorphisms or SNPs. So it depends on the type of goal or sequencing. Now, what are the objectives, learning objectives here? So I want to give you the knowledge for the reference-based and de novo assembly, and then the pipelines. Of course, I will also go for separate lectures and the advantage, disadvantage of reference and de novo assembly. Then, which method to use in different projects contexts. Then you can also go for the common tools and metrics for assembly quality. So first thing you need to know is what is genome assembly. Here, it is the computational process for stitching together short overlapping DNA sequence reads to reconstruct the original genome sequence. It reverses the sequencing process by reassembling the fragments into longer contiguous sequences that is called contigs, which are then linked into a scaffolds, forming a digital representation of the whole genome or of the genome. This is foundation or foundational step in bioinformatics for understanding genetic information. So here this is the genome and then this is the reads. But after assembly, it will be like this. Of course, there might be slight difference from the original reference genome to the reassembled genome. Now, key aspects of genome assembly is the process, then de novo assembly, computational tools, and importance of coverage. The fundamental challenge is to computationally infer the original order of the short DNA sequences since this is not known. This is achieved by finding overlaps between the reads where the suffix of one read matches the prefix of another to build a consensus sequence. So, de novo, then assembly, this refers to building the genome from scratch without a reference sequence by comparing and overlapping all the reads. Then, of course, it comes the most important part with which computational pipeline or softwares you are going to use. And what is the importance of coverage here? The high coverage is necessary to ensure there are enough overlapping reads to make confident connections between the fragments. A typical goal is 30 to 50x coverage. Now, what are the challenges? Challenges, repeats and heterozygosity, regions of DNA that are repetitive pose insignificant. It is basically posing a significant challenge, especially if the repeat sequences are longer than the sequencing reads. Heterozygosity, high heterozygosity genomes can lead to fragmented assemblies as reads for different alleles might be too different to be assembled together. Now, output and quality control. The output is said to be is a set of contigs and scaffolds representing the reconstructed genomes. Quality control is performed to assess the accuracy and completeness of the assembly. Then applications, a completed genome assembly is used for many downstream analyses, including gene annotation, comparative genomics, and functional genomics. Assemblies are often submitted to the public databases like the European Nucleotide Archive or NCBI, then there are some key terms which you need to remember while you are working on de novo genome assembly. What is read, what are pair int, make pair, contig, scaffold, coverage, what is 30x coverage and 60x coverage. Then KMR, which is most important in de novo genome assembly, and why the people need to use odd gamer number instead of even number, and then overlap layout consensus, and then of course reference. So paired end sequencing, how it works. So two reads are generated from opposite ends of a single sorted DNA fragment. Then insert size is smaller, typically between 300 and 500 bases or base pairs, best for improving the accuracy of alignment across repetitive regions of the genome, filling small gaps in the genome assemblies. Then make pair sequencing. How it works, two reads are generated from opposite ends of a much larger DNA fragment, and this is achieved by using a specialized library preparation that can create larger circular DNA fragments before sequencing. And the insert size here is larger, ranging from 2 to 20 kilobase. And it is best for span large repetitive regions that are difficult to assemble using short reads alone. Then detecting larger structural rearrangements like insertions, deletions, and inversions over longer distances. So pair end and mate pair are both DNA sequencing techniques that sequence two ends of a DNA fragment to provide information about the physical distance between them. But mate pair uses a much larger insert size compared to the paired end. This longer insert size makes make pair more effective for spanning large repetitive regions and detecting large structural rearrangements like inversions or translocations. Both the methods could improve genome assembly and accuracy by using the known distance between the two reads to link them, which helps to resolve the gaps and ambiguities that single reads cannot. Now, combining both short insert and pair int and long insert make pair data can maximize the information and lead to more contiguous and accurate genome assemblies. These short insert reads can fill in the gaps between the large structures identified by the mate pair reads. Now, what is contig? A contig is a continuous gapless stretch of DNA sequence, while a scaffold is a larger sequence that is formed by linking contigs together with the gaps of known or unknown length. Scaffolds provide a more complete picture of the genome by ordering and orienting the contigs, often using information from mate pair sequencing to connect them into longer structures that resemble the chromosomes. Now, you have already identified the definition of contigs, which is a continuous sequence of DNA assembled from overlapping fragments, and the composition is, of course, ATGC. Then role, it represents a single unambiguous segment of the genome. A scaffold, a longer sequence built by joining the contigs together. And composition, again, made up of multiple contigs, which is made up of ATGC by the gaps, connected by the gaps. These gaps are often represented by a series of M's in a sequence file. Now, role, it provides a framework for the genome by ordering and orienting the context, helping to reconstruct larger chromosomal structures. How they relate. So, building blocks. Contigs are the fundamental building blocks, and scaffolds built them from them. Assembly, genome assembly processes arrange reads into contigs. Then these contigs are linked into scaffolds with the ultimate goal of creating complete chromosomes or chromosomal arms. Gaps are the missing sequence information between the contigs within a scaffold. Advances in sequencing technology, such as long read sequencing, are helping to reduce the number and size of these gaps, leading to more complete assemblies. Now, what is coverage, 30x or 60x? So don't be confused with the coverage of microscope, because there also this term is used, but that is different. There you think for the magnification, but here you are thinking something for the depth of the coverage. So in genomics and DNA sequences, the coverage, also known as the depth of coverage or sequencing depth, refers to the number of times a particular nucleotide in the genome has been sequenced during a DNA sequencing project. And 30x, 60x, these figures indicate the average number of times each base in the genome has been read. For example, 30x coverage means that on an average, each base pair was sequenced 30 times. So the purpose is accuracy and variant detection. Now, it comes to Kramer or divergent graph and overlap layout consensus. So here from this figure, you can easily understand what we were defining the terms for the de novo genome assembly. So a chimer is the substring of a DNA or protein sequence with a fixed length denoted by K. These short sequences are used in bioinformatics to analyze large data sets by breaking them down into manageable pieces for tasks like genome assembly, sequence comparison, and identifying repetitive regions. So here, figure A is basically showing in figure A, step 1, it shows identify the reads that have overlap. And here you can see the reads, how it looks like. And then you go for sorting the reads, read A, read B, C, and D. So then you go for building the context. So in the third step of figure A, you can see the context built from the reads. Now, you go for figure B, where you can see Kramer processing. Here the Kramer size equals K equals 3. So you can see the read A, B, and C. But here you can see the three bases together. And that is here what it shows, the Kramer processing. And then you can go for the graph which I was discussing about divergent graph. And finally, you proceed for building the context, and then the context is here. Now, reference-based genome assembly or mapping assembly. When you have an existing high-quality reference genome, you map the new reads to it. Then identify the differences or variants, sometimes fill gaps or reassemble the segments. Typical setting, the resequencing of a known species or strain. Diagram reads mapping to reference, and then consensus sequence or variant calling. But when you are going to implement, for example, if you are going to work on RNA-seq by using reference-based genome assembly, then it is slightly different from the text as you can see in the figure. So you receive the FASTQ file, and then you proceed for quality check, trimming, and quality control. And then you proceed for the mapping, where you go for towards the genome or the transcriptome, and where you are using the BOTI to software or some alternate of it. Then you have some kind of annotation part where you can annotate the genes for each map, and then you go for if you are looking for the gene expression or differentially gene expressed or mutation, then you have some different steps. But in case of RNA-seq, once you identify or map, annotate the genes and you have the gene expression value, then you proceed towards differential gene expression profiling and pathway enrichment analysis. But if you are working on whole genome sequencing or whole exome sequencing or ataxic or chipseq data, then you have slightly different steps further. That's overall thing what you need to understand. And please don't forget about the Biomart ensemble database, which will be the major source of the files which you will need while you are performing the genome assembly. For example, the reference genome or the annotation file or GTF files and many more things. You will get direct access without any login or anything. It is free of cost, and that is the source present here in Ensemble Biomart. Of course, in addition to the reference genome or annotation file, there are many more application of Biomart Ensemble, but here our concern is for the reference genome, annotation files, and some other supplementary files for our purpose. Now, as I mentioned, that in reference-based genome assembly, the pipeline is quite simple. So quality check, you can say the first step where you can go for trimming adapters and filtering, aligning the reads, then process the alignments, sorting and marking the duplicates. Then variant calling, consensus generation, and then optional is fill the gap, structural variant detection, and further more things. Then, when you use reference-based assembly, good reference genomes are available and closely related to sample. So there are some exception cases also when you do not have the known reference genome, but you know that there are some closely related organism. So you could also map your sequencing file against the closely related organism. So that is also a kind of reference-based genome assembly. Then in reference-based assembly, less novel genome content is expected, lower cost, faster turnaround, and suited for resequencing population genomics. Now, what are the limitations of reference-based genome assembly? Missing novel sequences not present in the reference bias, structural variants may be missed or misrepresented, poor for highly divergent or new species, of course. Then we proceed for de novo genome assembly. So we have already defined the building genome sequence from scratch without relying on the reference genome. And reads are assembled into contigs by overlaps or graph methods. Diagram here, what is showing here is basically reads, then assembly graph, contigs, scaffolds, and genome. And all these terms have been defined in the previous slides. So what you need to understand in de novo genome assembly that you have the organism, but you don't know the reference genome, or you are not familiar that their reference genome is in good quality, then you need to proceed for de novo genome assembly. As you can see here in the figure, that there is some plant organism, and then here is the DNA, and then you go for whole genome sequencing, and again you build the reference genome because you are not having the whole genome sequence available for that specific purpose. And what are the steps which you can see in this figure? So remove low quality and contaminant reads, de novo assembly of the contigs, find closest reference to contigs, then merge overlapping contigs and fill the gaps, generate consensus genome, and identify the minority variants, such as substitution, insertion, deletion, and something more. So these are the steps which you have seen. So basically, there are 5 to 6 steps needed in general for de novo assembly, that is quality check, assembler, and constructing the context, scaffold, gap filling or polishing, and assembly quality check, and then finally you should have some kind of flow chart in your mind while you are working. Now, when to use the de novo assembly? As I mentioned, when there is no good reference genome, then studying new species, strain, or metagenome, expect novel genomic content, want a genome assembly rather than just variant calling. And then the limitations of de novo genome assembly, it requires higher coverage, better and quality, and then more computational. It is more computational intensive, that is in terms of RAM and CPUs. For example, for reference-based genome assembly, you can use the 16 GB RAM also. But while you are working on de novo genome assembly, irrespective of the file size, you need to have minimum 24 GB RAM, and then you can. And of course, CPU will decide how fast your calculation will go on. Then risk of fragmented assemblies if genome is complex. Then downstream annotation may be needed, of course. Then key challenges in genome assembly are repeats, which is the biggest challenge, heterozygosity or polyploidy, which complicates the assembly graphs, sequencing errors and coverage bias, choice of technology short or long reads, computing resource constraints. Now, the basic difference between reference-based and de novo genome assembly. In reference-based, need for reference, of course, yes, but in de novo, no. Novel sequence detection, limited, but in de novo, good if data quality is adequate. Computational cost, much lower than de novo, and best for reference genome-based genome assembly is best for resequencing and variants, but de novo is best for new species, strands, or fully assembly. Major risk in reference genome is reference bias, missing sequences, but in de novo, it is fragmentation and misassembly. Then the tools and software for both de novo and the reference-based genome assembly. Of course, these are some of them, but when I will be discussing or presenting the separate case studies or a real application of reference-based genome assembly and analysis, then I will be discussing for both of them. So, what you need to understand here in de novo assembly, some of the notable points: N50, L50, their definition and meaning, number of contexts, scaffolds, because you need to go for plot while you are performing the analysis, coverage of expected gene sets, percentage of genome aligned to the reference or related species, SS misassemblies. So N50 is the length of the shortest contig that covers at least 50% of a genome assembly, while L50 is the number of contigs that are needed to reach 50% of the assembly's total length. Both are used to measure the continuity and quality of a genome assembly, with a higher N50 and lower L50 generally indicating a better assembly. This is what you need to keep in your mind. Now, case study I will present for each type of reference-based genome assembly as well as de novo assembly. For example, I will cover RNA-seq analysis and whole genome, whole exome, chip-seq, attack-seq. And then, of course, similarly, the case study for de novo, I have already presented the metagenomics, but still I will again prepare it and present and meta transcriptome, where I will be showing the real application of de novo genome assembly. Now, choosing the right approach and decision workflow. That is very good. While you are familiar with all the points which I have discussed about de novo and reference-based assembly, then start with some of the questionnaire. Is there a good high-quality reference genome available? How divergent is my sample from the reference? Do I expect novel genomic content? What read types and coverage do I have? What computing resources are available with you? Then, based on answers, choose reference-based or de novo or hybrid approach. That is also possible. So, hybrid approach is basically sometimes a combined strategy is used. That means de novo assembly plus alignment to reference genome or reference-based plus local de novo for divergent reasons and discuss the advantages and when to use them. So hybrid genome assembly is a method that combines short and long DNA sequencing reads to create a more accurate and contiguous genome sequencing than either method can achieve alone. Short reads provide accuracy, while long reads provide the longer span needed to resolve the repetitive regions and connect distant parts of the genome. This approach is particularly useful for complex genomes and allows for the generation of high quality closed genomes for applications in research and conservation. So, next is the practical tips and best practices. Always check the quality before assembly. Choose sequencing technology and library strategy that matches your genome complexity. For de novo assembly, higher coverage and longer reads help to overcome the repeats. For reference based, ensure the reference is appropriate and account for divergent. Validate your assembly, map reads back, use quality check tools, check for contamination. Document parameters, versions, and record everything. Perform the annotation for de novo with the validated gene sets if required. So the common pitfalls and how to avoid them. Low coverage, fragmented assembly. High error rate reads without correction, misassembly. Then high repetitive or heterozygous genome accounted to poor results. Using too distant a reference, bias or incorrect mapping, not performing quality check at each step. So these things are the common pitfall you need to avoid. Now, future trends and considerations, emergence of ultra long reads, for example, from Oxford Nanopore, and their effect on de novo assembly, pan genomes and graph-based references, moving beyond single linear reference, single cell and metagenome assemblies, better algorithms, graph convolutional networks for assembly, that is emerging research. So in summary, you can recap all the things what we have discussed. For example, de novo, when you need to start from the scratch, when there is no reference genome available. So then the basic pipeline, and in reference genome, you have already the known genome sequence for the organism. Then key take home, choose the method that matches your data, goals, and resources. And good practice is always validate and report matrix clearly. So reference-based and de novo genome assembly, you need to memorize or repeat these things in your mind. What is the difference between them, when and where, and how to use these two assembly methods. If both of them are not suitable, then is it possible to use hybrid genome assembly? That means the combination of both of these methods. Now, you can also ask the questions from my side by putting the comments on my YouTube videos, and I will be happy to answer. And mostly, I provide quick answers for all the questionnaires posted on my video comments. So, further reading and resources, you can go for the F1000 research or Galaxy, that is also called use Galaxy, where you can find a number of or a large number of related references. Or if you are not familiar with these two things, then simply go to PubMed and search the keywords for reference-based genome assembly and the de novo genome assembly, and you will get all the information required for your family. So thank you very much for listening my videos, and in the future I will continue with all these common practices which every master's, bachelor, PhD students or research people are using such methods in daily life. Thank you very much.